You see that fuzzy patch on your sandwich, green dots spreading across yesterday's bagel, blue-gray fuzz claiming territory on toast you forgot in the back of the pantry. Everyone knows moldy bread, but what's actually growing there? What's that black stuff versus that white cottony mass? Which one makes you sick, and which one made penicillin? The grocery store tells you preservatives keep bread fresh. Your grandma says, just cut the mold off. Scientists tell you something different, something documented. Something with names and numbers and microscopic invaders that have been colonizing carbohydrates for millions of years. Welcome to the truth about what's eating your bread. If you value documented mycology and food science history like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. This script is researched and factual, using sources from the National Center for Biotechnology Information's PubMed Central Database, where we pulled mycotoxin contamination studies published in 2024 by research teams at the University of Natural Resources and Life Sciences Vienna and the European Food Safety Authority's comprehensive technical reports on aflatoxin contamination in cereals, last updated in their 2023 assessment cycle. Now back to the information. Rhizopus stolonifer, black bread mold, the one that shows up first, the one everyone's seen. When you leave bread out for 5 to 7 days, this is the colonizer. The genus Rhizopus exists on every continent on Earth. It's been documented since at least 1843 when French bakeries reported mysterious infestations. The mycelium starts white or pale blue-green, fuzzy and spreading. Then the center darkens. Black sporangia form at the tips of aerial hyphae, those vertical stalks reaching up from the bread surface. Each sporangium holds thousands of spores ready to launch into your kitchen air. The mold looks fuzzy because it's actually a network of stolons, horizontal runners that spread across the bread like highways, and rhizoids, root-like structures that burrow down into the starch, anchoring the colony and releasing digestive enzymes. Researchers at Kansas State University measured the growth rate in laboratory conditions. At 30 degrees Celsius, that's 86 Fahrenheit, Rhizopus spreads at approximately 4 millimeters per hour. In optimal conditions on wheat bread, a single spore can produce a visible colony covering 5 square centimeters in 72 hours. The thermal death point sits at 60 degrees Celsius. Below that, it survives. The mold grows in acidic environments with a pH as low as 2.2, which is why it thrives on sourdough as easily as white bread. Rhizopus stolonifer produces ethanol as a metabolic byproduct. Industrial operations harvest this. The mold converts sugar to ethyl alcohol at measurable rates, and companies use Rhizopus species to manufacture fumaric acid and high-purity lactic acid. But there's a darker side documented in medical literature since 1962. Rhizopus microsporus, a close relative, carries bacteria inside its cells, Burkholderia bacteria. These endosymbionts produce rhizoxin, a compound that causes rice seedling blight. A study published in Applied and Environmental Microbiology in 2009 by researchers Gabriel Lackner and Niels Mobius traced the toxin production. When scientists removed the bacteria from Rhizopus microsporus, the fungus stopped making toxin. The bacteria controlled reproduction too. Without its bacterial passengers, Rhizopus microsporus struggled to produce asexual spores, those greenish-black conidia that give the mold its color and allow it to spread. Rice accounts for 20% of calories consumed worldwide. This symbiosis threatened food security in regions dependent on rice cultivation. In humans with weakened immune systems, Rhizopus stolonifer becomes an opportunistic pathogen. The disease is called zygomycosis. It invades sinuses, causes respiratory infections, triggers automycosis, fungal ear infections. The action of smelling spoiled bread can expose you to spore inhalation. A case report from 1962 documented neurospora growing in the inner eye following cataract surgery. Wrong genus, but same lesson. Operating rooms needed to be spore-free. Food handlers needed to understand contamination vectors. Let's talk preservation. The bread industry discovered calcium propionate in the mid-20th century. Add 0.3% calcium propionate to dough, and Rhizopus colonies take 14 days to appear instead of 5. Sodium acetate works similarly. Modern toast breads list these on labels. Preservative-free artisan loaves from bakery sections mold faster because they lack these inhibitors. Research published in Food Production Processing and Nutrition in 2019 showed that modified atmosphere packaging extended bread shelf life to 24 days at 20 degrees Celsius when the atmosphere contained 60% carbon dioxide and 40% nitrogen. The high carbon dioxide retards germination. Low oxygen levels prevent aerial growth. But Rhizopus has one more trick. The bread matrix contains thousands of tiny pores that trap oxygen even in modified atmosphere packaging. Companies started adding ethanol emitters and oxygen absorbers inside packages. 1% ethanol released from an absorbent pad extended white bread shelf life beyond 60 days at 22 degrees Celsius, while control loaves developed chalk molds, visible colonies, within 10 days. These numbers come from studies by Ligon and Voisey, published in 1991 and updated in subsequent research through 2023. Penicillium, the blue-green colonies, the ones with the white fuzzy border, the genus that gave us the first antibiotic. There are multiple Penicillium species that colonize bread. Penicillium crustosum shows up frequently. So does Penicillium bialoizense, Penicillium paxillin, and the most studied one, Penicillium citrinum. These molds appear as fuzzy patches, colored white, gray, or light blue. Unlike Rhizopus, many Penicillium species grow in cold temperatures. Your refrigerator won't stop them. Optimal growth occurs between 5 and 25 degrees Celsius. The colonies spread in circular or irregular patches, powdery in texture, releasing clouds of conidia when disturbed. Alexander Fleming discovered penicillin in 1928 from Penicillium notatum growing on a Staphylococcus culture plate. By 1942, mass production began. The antibiotic molecule inhibits bacterial cell wall synthesis. It revolutionized medicine, saved millions in World War II. But penicillium on bread isn't necessarily producing penicillin. Different species make different compounds. Some penicillium molds create mycotoxins. Roquefortine-C appears in penicillium spots on bread. Researchers using DNA barcoding identified Penicillium crustosum and Penicillium paxillin on whole loaf cornbread in a controlled contamination study published in PMC in 2024. The study placed bread samples under multispectral imaging with 30 micrometer resolution per pixel. Each pixel captured surface reflection, enabling determination of mold area. The team from the University of Natural Resources and Life Sciences Vienna inoculated bread samples with known species and tracked mycotoxin migration. Roquefortine-C remained concentrated at the inoculation site. It didn't diffuse far. Fumonisin B2 and viridite toxin showed similar containment, but there's an exception we'll hit later. Penicillium molds also produce ochratoxin in certain conditions, though this is more common in Penicillium verrucosum growing on grain during storage. Ochratoxin A is a nephrotoxin, a kidney poison, and a probable human carcinogen according to the International Agency for Research on Cancer. Maximum levels are regulated in the European Union at 5 micrograms per kilogram for cereal products. Blue cheese uses Penicillium roqueforti deliberately. Gorgonzola, Roquefort, Stilton, all rely on controlled penicillium growth for flavor and texture. The blue-green veins running through the cheese are fungal hyphae. The compounds they produce create the sharp, tangy taste. Cheesemakers inject Penicillium roqueforti spores into cheese curds, then age them in caves or climate-controlled rooms with specific humidity and temperature ranges. The mold requires oxygen to grow, so cheesemakers pierce the cheese with needles to create air channels. After 90 days, the veins are visible. After 6 months, the flavor peaks. But Penicillium roqueforti on bread is different from Penicillium roqueforti in cheese. The substrate matters. The metabolites produced vary with available nutrients. Bread contains different sugars and proteins than cheese curds. The mold adapts, On bread, Penicillium species might produce mycotoxins not present in controlled cheese production. The difference is regulatory oversight and strain selection. Cheese producers use specific water-tested strains. Bread contamination involves wild strains with unpredictable metabolite profiles. Aspergillus, the dark mold, the genus that makes aflatoxin, one of the most potent carcinogens known. Aspergillus niger appears black. Aspergillus flavus shows up yellow tan. Aspergillus parasiticus produces aflatoxins B1, B2, G1, and G2. These compounds are diffuricoumarin derivatives. Identified and chemically characterized in 1963, aflatoxin B1 is the most toxic, classified as Group 1 carcinogen by IARC. It causes hepatocellular carcinoma, liver cancer. The mechanism involves DNA damage and mutation of the p53 tumor suppressor gene. When Aspergillus flavus or Aspergillus parasiticus infect crops before harvest or contaminate grain during storage, the toxins accumulate. Warm conditions, 27 to 30 degrees Celsius, and high moisture favor aflatoxin production, 7% moisture content minimum. The fungi grow on bread, peanuts, corn, wheat, rice, cottonseed, tree nuts. A study published in Mycopathologia in 1982 by researcher J. Rice tested 3 Aspergillus strains on bakery products: Aspergillus flavus NRRL 3251, Aspergillus parasiticus CMI 89717, and Aspergillus parasiticus NRRL 2999. Only the last strain produced aflatoxin M1 on bread substrates. The highest yield was 4 micrograms per kilogram on whole wheat bread at a total acid content of 5 to 6 and incubation temperature of 30 degrees Celsius. The toxin levels decreased with longer growth time, probably due to further metabolism of aflatoxin M1. The fungus was converting the toxin to other compounds or sequestering it, but 4 micrograms per kilogram matters. The FDA set the first regulation on aflatoxins in 1965 at 30 micrograms per kilogram. Modern limits are stricter. The European Commission regulation from 2006 sets maximum levels at 2 micrograms per kilogram for aflatoxin B, 1 in cereals and cereal products intended for direct human consumption. In 1961, 100,000 turkeys died in England from Turkey X disease. Investigators traced it to aflatoxin-contaminated peanut meal and feed. That launched mycotoxicology as a scientific field. Then came Kenya in 2004. An acute outbreak of aflatoxicosis from contaminated maize killed 125 people. Symptoms included high fever, vomiting, ascites, fluid accumulation in the abdomen, liver failure, jaundice. Mortality rate hit 60% in severe cases. The Soviet Union experienced mass casualties in the 1930s and 40s when fusarium-contaminated wheat flour was baked into bread. The disease was called alimentary toxic aleukia. Symptoms started with abdominal pain, diarrhea, vomiting, prostration. Within days came fever, chills, myalgias, bone marrow depression, granulocytopenia, secondary sepsis, then pharyngeal and laryngeal ulceration, diffuse bleeding into skin, melena, bloody diarrhea, hematuria, hematemesis, epistaxis, vaginal bleeding, pancytopenia, gastrointestinal ulceration, mortality 60%. Fusarium sporotrichoides contamination was found in affected grain in 1932. The active ingredient was trichothecene T2 mycotoxin. It was produced in quantity and weaponized before the Biological Weapons Convention passed in 1972. But back to Aspergillus on bread. Aspergillus pseudoglottis was identified on wheat rye whole loaf bread in the Vienna study. Aspergillus tubangensis appeared on cornbread. These species produce different metabolite profiles. Aspergillus niger synthesizes malformin C, detected in the Vienna research using high-performance liquid chromatography coupled with mass spectrometry. Malformin C is a cyclic pentapeptide with phytotoxic properties. It causes plant cell malformation. In animals, toxicity data remains limited. But the compound is structurally similar to other bioactive peptides with potential health impacts. Aspergillus species also produce ochratoxin A, particularly Aspergillus ocraceus. This mycotoxin appears in coffee, dried fruits, wine, beer, and grain. It's nephrotoxic and hepatotoxic. Studies in rats and mice proved carcinogenicity. Human epidemiological studies linked ochratoxin exposure to urinary tract tumors, though confounding factors complicate the data. Aspergillus carbonarius is the main species found on vine fruit, releasing toxin during juice making. The European Food Safety Authority published technical reports on ochratoxin levels in cereals and issued recommendations in their 2023 assessment. Maximum levels sit at 3 micrograms per kilogram for all products derived from unprocessed cereals, and 0.5 micrograms per kilogram for processed cereal-based foods for infants and young children. Now the baking process. Does heat kill mycotoxins? Short answer, no. Aflatoxins are thermostable. A study published in 2007 examined the effect of bread-making steps on aflatoxin degradation. Researchers inoculated 2 wheat cultivars, Saka 8 and Gemisa 5, with Aspergillus flavus alone and combined with Aspergillus ocraceus. They measured aflatoxin at whole grain stage, after milling, after fermentation, and after baking. The highest reduction percentage for total aflatoxins occurred in Saka 8 single treatment at 32.96%. After baking, aflatoxin B, 1 reduced by 31.98%, aflatoxin G, 1 by 44.53%, aflatoxin G, 2 by 35.35%, and total aflatoxins by 41.17%. That means 58 to 68% of the toxin remained in baked bread. Mycotoxins don't just burn away. They persist through commercial baking temperatures of 190 to 230 degrees Celsius. The compounds are chemically stable under those conditions. Cladosporium, the dark green to black mold. Large, round, fairly smooth colonies. Strong odor, the genus most irritating to people with mold allergies. Cladosporium species cause sneezing, coughing, wheezing on prolonged exposure. They're airborne allergens. common in outdoor environments, but they colonize bread readily. Cladosporium remitinellum was identified in the Vienna bread study using DNA barcoding. It appeared on wheat rye bread and corn bread, growing alongside Aspergillus and Penicillium species. Cladosporium sphaerospermum is another bread colonizer. These molds appear dark because of melanized cell walls. The pigment protects against UV radiation and environmental stress. The colonies spread as circular patches, smooth-surfaced compared to the fuzzier Rhizopus and Penicillium. Under microscope, Cladosporium shows branching conidiophores with chains of oval conidia. Cladosporium doesn't produce the high-risk mycotoxins like aflatoxin or ochratoxin, but it does generate metabolites that trigger allergic responses. People with asthma or compromised respiratory systems react to Cladosporium spore inhalation. The mold releases volatile organic compounds, VOCs, that have musty odors. These VOCs include terpenes, alcohols, ketones, and aldehydes. Some are irritants. Some are potential allergens. Research on indoor mold published in Scientific World Journal in 2013 documented that Cladosporium is among the genera associated with respiratory inflammation in buildings with water damage. The spores are ubiquitous in air, with concentrations fluctuating by season, even hour to hour. When bread sits out, Cladosporium spores land and germinate if moisture is adequate. The mold prefers slightly lower temperatures than Aspergillus, growing well at 15 to 25 degrees Celsius. It's common in refrigerated bread, which is why even cold storage doesn't guarantee prevention. If you value documented mycotoxin research and fungal biology like this, make sure you're subscribed. It helps us keep producing detailed science content. Comment what weird mold you've seen lately. This research comes from peer-reviewed sources including PMC's database of over 11,000 mycotoxin contamination studies indexed through 2024. And specific papers like the one from the team at the University of Natural Resources and Life Sciences Vienna, published in Foods Journal in July 2024, analyzing bread mold metabolite distribution using multispectral imaging and HPLC-MS techniques. Now, back to the story. Neurospora crassa, the so-called red bread mold. Though scientists dispute that name, Neurospora produces orange pigment, not red, except for a brief transitional stage. The first documented case was in French bakeries in 1843. The pathologist Anselme Payen reported extraordinary alteration of munition bread. He used the terms champignon rouge and champignon de couleur rouge-orange, red fungi and red-orange fungi. Neurospora belongs to the phylum Ascomycota. It forms broadly spreading colonies with abundant ascomata, fruiting bodies. The characteristic feature is striations on the spores, which look like nerve fibers under microscope. The name Neurospora means nerve spore in Greek. When dark-grown cultures are first brought into light, they're colorless. Then within an hour, they become pigmented. The initial blush is pink or red, but it quickly transforms to orange. Since the ephemeral red stage is rarely seen, most observers see orange or yellow-orange mold. This caused confusion in naming. By 1927, Don Hotz, C. L. Shear, and B. O. Dodge described the sexual phase and named the genus in a classic paper. Neurospora crassa became a model organism for genetics. In 1941, George Beadle and Edward Tatum at Stanford University used it for Nobel Prize-winning work. They exposed Neurospora crassa to X-rays, causing mutations. Then they observed failures in metabolic pathways caused by errors in specific enzymes. This led to the one gene, one enzyme hypothesis, that specific genes code for specific proteins. The hypothesis was later refined to one gene, one polypeptide, acknowledging that not every protein is an enzyme, but the work founded biochemical genetics and launched molecular genetics. The genome of Neurospora crassa was fully sequenced in 2003. It's 43 megabases long with approximately 10,000 genes, published in Nature by a consortium led by J. E. Galligan. Neurospora has been used in research on circadian rhythms, epigenetics, gene silencing, cell polarity, DNA and RNA studies, sugar metabolism. Its circadian rhythm runs on a 22-hour cycle, influenced by light and temperature. But is Neurospora dangerous? Not really. It's neither toxic nor pathogenic to humans, animals, or plants. There are no known cases of home contamination causing disease. Unlike Aspergillus or Penicillium, Neurospora doesn't produce mycotoxins. It's an obligate aerobe, unable to grow in the gut, bladder, tissues, or systemically. Medical mycology textbooks either fail to mention it or list it only as a potential laboratory contaminant. One 1962 report cited neurospora growing in the inner eye following cataract surgery, but that's an adventitious infection requiring surgical breach and contamination, not normal exposure. Neurospora cytophylla, the asexual form once classified as Motilia cytophylla, was a common bakery contaminant before fungal growth inhibitors and improved hygiene. The mold was difficult to eradicate and caused financial losses. Now it's rare on commercial bread. Neurospora is more commonly found on sugarcane bagasse, in soil, on tree bark, especially burned trees after forest fires. The germination of its spores is triggered by thermal shock. After wildfires, neurospora blooms appear on burned stubble and grass. Dense growth can blanket many acres when filter mud from sugar refineries is spread on fields. The orange mold is conspicuous, spreading rapidly. Fusarium, the genus that produces trichothecenes, fuminizins, zearalenone, plant pathogens, crop destroyers, and producers of some of the most toxic compounds in agriculture. Fusarium species infect wheat, maize, barley, rice, oats during growth or after harvest. Fusarium head blight, also called scab or ear disease, is a worldwide problem. It causes yield loss and reduced grain quality. When infected grain is milled into flour and baked into bread, the mycotoxins persist. Fusarium graminearum produces deoxynivalenol, called DON or vomitoxin. It inhibits protein synthesis in eukaryotic cells. In animals, it causes feed refusal, vomiting, diarrhea, weight loss, immune suppression. In humans, chronic exposure is linked to growth retardation and immune dysfunction. The European Union set maximum levels at 750 micrograms per kilogram for DON in bread, pastries, cakes, and biscuits. Fusarium species appear pink on barley and wheat when contamination is extreme. The mycelium spreads as white to pinkish masses on grain heads. On bread, Fusarium colonies are less visually distinct than Rhizopus or Penicillium, often appearing as diffuse whitish or pale pink patches, but the mycotoxins are the real threat. T2 toxin and HT2 toxin are type A trichothecenes produced by Fusarium sporotrichoides and Fusarium langsithiae. These are more toxic than DON. They cause alimentary toxic eleukia, the disease that killed an estimated 100,000 people in the Soviet Union between 1942 and 1948. Symptoms included hemorrhaging, necrosis, immune collapse. The trichothecene mechanism involves inhibition of protein synthesis, DNA damage, lipid peroxidation, disruption of cell membrane integrity. T2 toxin binds to the 60S ribosomal subunit, blocking peptidyltransferase activity. And without protein synthesis, cells can't repair damage or maintain function. The toxic dose varies by species. Lethal dose for ducks is lower than for chickens, lower than for rabbits. Humans are sensitive, but exact LD50 data is unavailable for ethical reasons. Fumonisins are another Fusarium product. Fumonicin B1 is produced mainly by Fusarium verticillioides and Fusarium proliferatum, which contaminate maize. Fumonicins disrupt sphingolipid metabolism. They inhibit ceramide synthase, causing accumulation of sphingoid bases and depletion of complex sphingolipids. This affects cell signaling, membrane integrity, apoptosis. In horses, fumonicins cause equine leukoencephalomalacia, a fatal brain disease. In pigs, they cause pulmonary edema and hydrothorax. In rodents, fumonicins induce liver cancer. Human epidemiological studies in regions with high maize consumption, like parts of South Africa and China, linked fuminicin exposure to esophageal cancer. The International Agency for Research on Cancer classifies fuminicin B1 as possibly carcinogenic to humans. Group 2B. During bread making, fuminicins remain stable. Milling has little effect, kneading, fermenting, and baking don't degrade them significantly. A study on the fractionation of Fusarium mycotoxins during maize milling found that bran ice, the outer layers discarded for white flour production, contain the highest concentrations. White flour has lower levels, but it's not zero. Zearalenone is a phenolic resorcilic acid lactone mycotoxin produced by Fusarium graminearum and other Fusarium species. It contaminates maize and other cereal grains. Zearalenone is an estrogenic compound. It binds to estrogen receptors, mimicking the hormone. In animals, it causes reproductive disorders. Pigs are particularly sensitive. Exposure leads to vulvovaginitis, reduced fertility, pseudopregnancy, abortions. In cattle and sheep, it affects reproductive performance and feed intake. In humans, chronic exposure raises concerns about endocrine disruption, though data is limited. The European Union set maximum levels at 75 micrograms per kilogram for bread, pastries, cakes, and biscuits. Zearalenone undergoes metabolism in plants, fungi, and animals. Modified forms include alpha-zearalenol, or CAOS, beta-zearalenol, and glucose conjugates. These masked mycotoxins can hydrolyze back to the parent compound during digestion, releasing free zearalenone in the gut. Meniscus, the genus that produces red pigments used in Asian food, and also produces citrinin, a nephrotoxic mycotoxin. Meniscus species have been used in China since the Tang Dynasty, 618 to 907 AD, to ferment red yeast rice, make wine, color food. Meniscus purpureus, Meniscus ruber, Meniscus aurantiacus, all produce orange to red pigments as secondary metabolites. These pigments belong to three main types: red, orange, and yellow polyketones. They're used as natural food colorants in China, Japan, Southeast Asia, but Meniscus also produces citrinin. The fungi of genera Penicillium, Aspergillus, and Meniscus are the major citrinin producers. Penicillium citrinum is most common, but Meniscus contributes to contamination in fermented products. Citrinin is a polyketide mycotoxin. Chemical formula C13H14O5. It appears as lemon yellow needles at pH 4.6. The color changes to cherry red at pH 9.9. It was first isolated from Penicillium citrinum in 1931. Citrinin is nephrotoxic. It targets kidneys at 50 micromolar concentration. It deregulates calcium homeostasis in pig kidney cells, leading to cell death. In kidney and liver mitochondria, citrinin inhibits activities of oxidoreductases and ATPases, interferes with the electron transport chain, reduces efficiency of mitochondrial phosphorylation, changes transmembrane potential. Intracellular respiration efficiency drops. Protease activity is blocked. The lethal dose varies by species. For ducks, 57 milligrams per kilogram. For chickens, 95. For rabbits, 134. Citrinin is associated with yellow rice disease in Japan. It acts synergistically with ochratoxin A to depress RNA synthesis in murine kidneys. In the Vienna bread contamination study published in 2024, citrinin was the exception. While fumonisins and roquefortine C remained localized near the mold spot, citrinin diffused throughout the bread. When Penicillium citrinum, or citrinin-producing Meniscus species, contaminated bread, the mycotoxin was detected in almost all tested bread areas at high concentrations. Citrinin is highly mobile in bread matrix. That means cutting off the visible mold doesn't remove citrinin. The toxin spreads beyond the visible colony, migrating through the porous structure. The researchers used multispectral imaging with 30 micrometer per pixel resolution and high-performance liquid chromatography, coupled with mass spectrometry to track citrinin distribution. They inoculated bread slices with Penicillium citrinum, incubated them, then sectioned the bread into zones: the inoculation spot, the area directly below, and lateral distances of 3 centimeters. Citrinin appeared in all zones. The study concluded that removal of moldy parts may reduce food waste for insect farming or animal feed. But citrinin contamination makes partially moldy bread unsafe for human consumption even after visible mold removal. Meniscus fermentation products are commercially available. Red yeast rice supplements contain Meniscus fermented rice, marketed for cholesterol lowering because Meniscus produces monocolin K, which is chemically identical to lovastatin, a statin drug. But citrinin contamination is a concern. A 1999 study published in Mutation Research analyzed commercial Meniscus samples using HPLC. Citrinin was detected in all samples at concentrations ranging from 0.2 to 17.1 micrograms per gram. The study also tested mutagenicity using Salmonella microsome assay and Salmonella hepatocyte assay. Citrinin and 2 Meniscus extracts induced dose-dependent mutagenic response in strain TA98. The European Food Safety Authority issued opinions on citrinin in 2012. The panel noted that common food-relevant citrinin producers include Penicillium expansum, Penicillium citrinum, Penicillium verrucosum, and Meniscus species. They recommended strategies to reduce citrinin in Meniscus products, including strain selection, fermentation control, and addition of flavonoids that inhibit citrinin production. Research published in 2020 showed that genistein, a soy isoflavone, reduced citrinin by 80% in Meniscus aurantiacus fermentation when added at 2 grams per liter during liquid state fermentation. Pigment and biomass increased by approximately 20%. Chaetomium globosum, a less common but documented bread mold. It's a soft rot fungus, cellulose degrader, appears as dark greenish-brown to black colonies. It produces metabolites including ketoglobosins, cytotoxic compounds. The Vienna study expected to find chitomium mycotoxins on bread, but didn't detect them, possibly due to weak growth on the tested bread types. Chitomium is more common on cellulose-rich substrates like paper, drywall, and wood. When it does colonize bread, it contributes to the musty odor and dark discoloration. Chitomium species are among the fungi associated with water-damaged buildings. They produce volatile organic compounds that smell earthy or musty. People exposed to chitomium in indoor environments report respiratory symptoms, headaches, nausea. But on bread, chitomium is a minor player compared to rhizopus, penicillium, aspergillus, mucor, another zygomycete like rhizopus, commonly found in soil, plant surfaces, decaying organic matter. Mucor appears as white to beige or gray colonies, fast-growing, fluffy texture. The sporangiophores are simple or branched, forming apical globular sporangia. Unlike rhizopus, mucor lacks stolons and rhizoids. The colonies spread rapidly, covering bread in 48 to 72 hours. Mucor species are generally considered less pathogenic than Aspergillus or Fusarium, but they can cause mucormycosis in immunocompromised individuals. The infection is invasive, affecting sinuses, lungs, brain, skin. Mortality is high without aggressive treatment. Mucor is also used industrially. Some species produce enzymes used in cheese making and other fermentation processes. On bread, mucor competes with rhizopus for territory. Both genera spread quickly, but rhizopus typically dominates. Alternaria, a genus known for producing Alternaria toxins. Alternaria alternata is a common environmental mold, black or dark green, forming velvety colonies. It grows on fruits, vegetables, grains, bread. Alternaria produces alternarial monomethyl ether, altenuine, tenuazonic acid, all mycotoxins with various toxicities. Alternarial and alternarial monomethyl ether are mutagenic and potentially carcinogenic. Tenuazonic acid inhibits protein synthesis, causes hemorrhaging in animals. Alternaria is more associated with outdoor crops and produce than bread, but it colonizes bread when spores are present. The mold prefers slightly damaged or overripe substrates. On bread, Alternaria appears as dark spots with fuzzy edges, releasing powdery conidia when disturbed. The spores are large, multicellular, with distinctive longitudinal and transverse septa visible under microscope. Stachybotrys chartarum, the infamous toxic black mold of water-damaged buildings, it produces satrotoxins and other trichothecenes. Stachybotrys is not a common bread mold because it requires very high moisture levels and cellulose-rich substrates. It grows on drywall, ceiling tiles, wood, not typically on bread, unless bread is stored in extremely humid conditions for extended periods. But when it does grow, Stachybotrys is dangerous. The mycotoxins are potent. Satrotoxin H and G are macrocyclic trichothecenes, more toxic than T2 toxin. They inhibit protein synthesis, cause immunosuppression, hemorrhaging, neurotoxic effects. Stachybotrys exposure is linked to respiratory problems, chronic fatigue, cognitive impairment in people living in water-damaged buildings. The mold requires water activity above 0.94 DAUS, which is higher than typical bread moisture. So while Stachybotrys is a serious indoor mold concern, it's not a major bread contaminant. Now the undesired contamination. The Vienna study encountered 6 undesired mold spots on bread samples during testing. These weren't the intentionally inoculated species. They came from environmental contamination during handling or packaging. DNA barcoding identified Aspergillus pseudoglottis, Penicillium crustosum, Cladosporium ramotinellum, Aspergillus tubangensis, Penicillium bioloviscens, and Penicillium paxillin. The identified mold types were not available in the laboratory's mold library, except Cladosporium ramotinellum, which hadn't been cultured in that lab within the last year. This indicates the species originated from the bakery or packaging. Bread was transferred immediately to laminar hood in packaging after purchase, handled with sterile equipment, stored in autoclaved plastic containers. Still, contamination occurred. Mold spores are ubiquitous. They float in air, land on surfaces, survive on equipment. The undesired mold spots showed specific metabolite patterns. Penicillium produced Roquefortine C. Aspergillus produced Malformin C. Even though the intentionally inoculated mycotoxins decreased with distance from the inoculation site, higher concentrations of the same mycotoxins were detected in the undesired contamination spots compared to unaffected regions. This raises a question. Is it safe to cut off the moldy part and eat the rest? For most mycotoxins, yes. The Vienna study concluded that metabolites were exclusively concentrated in the inoculation area and directly below. Lateral distance of 3 centimeters showed no significant contamination for fuminizins. Roquefortine-C, verditoxin, dihydrocitronone. The exception was citronin. When citronin-producing molds are present, the toxin diffuses throughout the bread. High amounts were detected in almost every bread aliquot measured, so the answer depends on the mold species. If it's rhizopus, penicillium that doesn't produce citronin, or cladosporium, cutting off the mold plus a generous margin probably removes most contamination. But if it's Penicillium citrinum, Monascus, or any citronin producer, the entire loaf is suspect. The problem is, you can't identify mold species by eye with certainty. Penicillium species look similar. Aspergillus species vary in color. Without DNA barcoding or microscopic examination, you're guessing. The bread industry addresses this with preservatives: calcium propionate, sodium propionate, sorbic acid, ascorbic acid. These inhibit fungal growth. Modified atmosphere packaging reduces oxygen, adds carbon dioxide. Ethanol emitters release vapor that suppresses spores. Ultraviolet irradiation delays germination. All these methods extend shelf life, reduce waste, prevent mycotoxin formation. But consumers want clean labels: natural, preservative-free, artisan bread. The trade-off is shorter shelf life and higher mold risk. Freezing works. Fungal growth requires liquid water at minus 18 degrees Celsius, standard freezer temperature. Mold can't grow. Spores survive but remain dormant. Bread can be frozen immediately after baking, stored for months, then thawed. This is why commercial operations often freeze bread for distribution. The downside is texture change. Ice crystals form, disrupting starch structure. Thawed bread can be slightly drier, less fresh tasting, but it's safe. What about antifungal compounds? Researchers have tested natural antimicrobials. Essential oils from mustard, cinnamon, oregano, thyme contain compounds that inhibit mold growth. A study in 2000 examined mustard, essential oil in modified atmosphere packaging for rye bread. 1 to 10 micrograms per milliliter in volatile phase was effective against Penicillium commune, Penicillium roqueforti, Aspergillus flavus, Endomyces fibuliger. High carbon dioxide levels retarded growth, but didn't completely stop it. The problem is, essential oils affect flavor. Bread with mustard oil doesn't taste like plain bread. Acceptable concentrations for antimicrobial effect may alter sensory qualities. So industry uses essential oils sparingly or in combination with other preservation methods. Lactic acid bacteria are another approach. Certain strains produce antifungal metabolites during fermentation. Sourdough bread, made with lactobacillus cultures, has longer shelf life than standard yeast bread. The lactic acid lowers pH, creating hostile environment for molds. Acetic acid, produced by some sourdough bacteria, also inhibits fungal growth. Traditional sourdough uses wild fermentation, capturing environmental bacteria and yeast. Modern commercial sourdough may use defined starter cultures with selected strains. The result is bread with pH around 4 to 4.5 compared to 5.5 to 6 for regular bread. That pH difference slows mold colonization by days. A study on modified atmosphere with 60% carbon dioxide and 40% nitrogen extended sourdough bread shelf life to 24 days at 20 degrees Celsius. Control bread molded in 7 days. But let's return to the human health question. What happens when you ingest mold or mycotoxins? Acute exposure to high doses causes immediate symptoms. Aflatoxin at 1,000 micrograms per kilogram or higher causes aflatoxicosis, high fever, vomiting, liver failure, edema, jaundice. Trichothecenes cause alimentary toxic aleukia, gastrointestinal distress, hemorrhaging, immune collapse. Fumonisins cause acute liver and lung damage in animals, less clear in humans, but most exposure is chronic, low dose. You eat bread with trace mycotoxins day after day, year after year. The compounds accumulate. Aflatoxin B1 is metabolized by liver cytochrome P 450 enzymes to aflatoxin B189 epoxide, a highly reactive compound that binds to DNA, causing mutations. The P53 gene is particularly vulnerable. Mutations in P53 disrupt cell cycle control, leading to uncontrolled proliferation. Cancer. Regions with high aflatoxin exposure in diet have higher rates of hepatocellular carcinoma. China, Sub-Saharan Africa, Southeast Asia. The IARC working groups used data on aflatoxin levels in food to relate dietary intake to liver cancer incidence. The correlation is strong. Ochratoxin. It targets kidneys. Chronic exposure causes chronic interstitial nephropathy, kidney fibrosis. It's been linked to endemic Balkan nephropathy, a disease affecting people in rural areas of Croatia, Bosnia, Serbia, Bulgaria, Romania. The disease causes progressive kidney failure. Ochre toxin contamination of grain in those regions is suspected as causal factor. Citrinin is also nephrotoxic. The two mycotoxins act synergistically, meaning combined exposure is worse than either alone. Fumonisins disrupt sphingolipid metabolism, affecting multiple organ systems. Chronic exposure is linked to neural tube defects in areas with high fumonisin levels in maize. Zearalenone, as an estrogen mimic, may contribute to endocrine disruption, though human studies are limited. The European Food Safety Authority continuously updates assessments based on new exposure data and toxicology studies. For people with mold allergies, the mold itself is the problem, not just mycotoxins. Inhaling spores triggers immune response. Immunoglobulin E antibodies bind to mold antigens. Mast cells release histamine. Symptoms include sneezing, runny nose, itchy eyes, coughing, wheezing. Asthma exacerbations can occur. Cladosporium, Alternaria, Aspergillus, Penicillium, all are common allergens. Skin prick tests and serum IgE assays can identify specific mold sensitivities. People with known mold allergies should avoid moldy environments and discard moldy food without handling it extensively. Wrap moldy bread in plastic before disposal to prevent spore release into air. The global burden of mycotoxin contamination is significant. The Food and Agriculture Organization reports 25% of crops worldwide are contaminated. More recent studies suggest 60 to 80% of crops have some mycotoxin presence, though levels vary. Climate change affects mold distribution and mycotoxin production. Warmer temperatures and changing precipitation patterns create favorable conditions for Aspergillus and Fusarium. Modeling studies predict increased aflatoxin contamination in European cereal crops as temperatures rise. The University of Piacenza completed a project for the European Food Safety Authority, using predictive models to map potential future contamination. Results showed northward expansion of aflatoxin risk zones. Countries previously unaffected may see aflatoxin issues in coming decades. Fusarium head blight is already increasing in wheat-growing regions due to wetter spring weather. This translates to higher Dion levels in flour. Economic losses are substantial. The United States estimated $3 billion in losses to barley and wheat crops from Fusarium between 1991 and 1996. Globally, mycotoxin contamination reduces agricultural productivity, increases food waste, drives up costs. Testing and monitoring programs add expense. Maximum residue limits require enforcement. Rejected batches mean lost income for farmers. But the cost of not controlling mycotoxins is higher. Human health impacts, livestock losses, trade restrictions. International trade in grain involves strict mycotoxin testing. Shipments that exceed limits are rejected, returned, or destroyed. Reputation damage affects exporting countries. Food safety is paramount. So, what do you do at home? Store bread in cool, dry conditions. Room temperature is acceptable if you'll consume it within a few days. Refrigeration slows mold growth but accelerates staling due to starch retrogradation. Freezing is best for long-term storage. If mold appears, assess the extent. A small spot on one slice in a loaf may not have contaminated the rest if it's rhizopus or aspergillus and the mold is recent. But if multiple slices show mold, or if the mold is extensive, discard the loaf. If the mold is producing a strong chemical or musty odor, discard it. If you're immunocompromised, pregnant, very young, or elderly, discard any moldy food without exception. Risk isn't worth it. For healthy adults, cutting off visible mold, plus a generous margin of at least 2 to 3 centimeters, might be acceptable for hard substrates like cheese. But bread is porous, hyphae penetrate easily. When in doubt, throw it out. The cost of replacing a loaf is minimal compared to the cost of mycotoxin exposure or fungal infection, but the revelation comes when you realize bread mold isn't just an inconvenience. It's a window into fungal ecology, evolution, toxin production, human microbe interaction. These organisms have been colonizing organic matter for millions of years. Fungi predate terrestrial plants. They enabled plant colonization of land by forming mycorrhizal associations. They decompose dead material, recycling nutrients. Without fungi, ecosystems would suffocate in undecomposed biomass, but fungi also produce compounds that protect their territory. Mycotoxins evolved as competitive weapons against bacteria and other fungi. Penicillin kills bacteria, giving Penicillium an advantage. Aflatoxin may deter insect herbivory or suppress bacterial competitors. Trichothecenes are phytotoxic, helping fusarium invade plant tissues. The compounds that harm us are incidental. We're not the target. We just happen to consume contaminated food. The breadth of mold diversity on bread reflects environmental spore load, storage conditions, preservative use, substrate composition, white bread versus whole wheat, with bran versus without, fermented versus not, sliced versus whole loaf, all influence which species dominate. Toast bread with preservatives grows mold slower. Preservative-free artisan bread molds faster. Cornbread showed different species than wheat bread in the Vienna study. Substrate chemistry matters. Different sugars, different proteins, different moisture content. Molds adapt to available nutrients. Rhizopus thrives on simple sugars. Penicillium can utilize a wider range of substrates. Aspergillus tolerates lower water activity. A study published in Applied Mycology and Biotechnology in 2004 traced the genomics of Neurospora crassa. 10,000 protein-coding genes, some shared with other eukaryotes, some unique. Light-sensing proteins for circadian rhythm regulation. G-protein-coupled receptors not previously identified in fungi. The genome sequence revealed biosynthetic pathways, regulatory networks. Evolutionary history. Similar work has been done on Aspergillus, Penicillium, Fusarium. Whole genome sequencing enables understanding of mycotoxin gene clusters. Aflatoxin biosynthesis involves a 70-kilo base DNA sequence with 25 genes. The pathway includes at least 23 enzymatic reactions and 15 intermediates. Aspergillus, Parasiticus, and Aspergillus flavus both contain the complete gene cluster. Fumonisin biosynthesis in Fusarium. Verticillioides involves a different set of genes. Researchers identify these clusters, then investigate regulatory mechanisms. What environmental signals turn on mycotoxin production? How do fungi sense nitrogen levels, pH, temperature, and moisture? What transcription factors control the genes? Can we disrupt the pathway? Biological control is one strategy. Toxigenic strains of Aspergillus flavus, strains that don't produce aflatoxin, can outcompete toxigenic strains in fields. They occupy the same niche, colonize the same crops, but don't make toxin. By applying toxigenic strains as biopesticides, farmers reduce aflatoxin contamination. This approach is used in the United States and parts of Africa. Products like Afligard contain a toxigenic Aspergillus flavus, spread in fields before harvest. These strains reduce toxigenic strain populations. Studies show reductions of 50 to 90% in aflatoxin levels in treated crops. The method is biological, doesn't require synthetic chemicals, and is sustainable, but it requires application at the right time, under right conditions, with right strain selection. Research continues to optimize formulations and delivery. Plant breeding offers another solution. Some maize lines are resistant to Fusarium ear rot. Resistance involves physical barriers like tight husk coverage and biochemical defenses like antifungal proteins and phenolic compounds. Breeding programs select for these traits. Genetic engineering adds genes that enhance resistance. Beet maize, engineered to produce Bacillus thuringiensis toxin against insects, indirectly reduces Fusarium contamination because insect damage creates entry points for fungi. Fewer insect wounds mean less fungal infection. Field trials show lower DON levels in Bt maize compared to conventional varieties. Regulatory approval varies by country. Europe is cautious. The United States, Brazil, and others have adopted Bt crops widely. Post-harvest interventions include drying, sorting, chemical treatment, irradiation. Grain moisture must drop below 14% to prevent mold growth during storage. In humid climates, mechanical dryers are essential. Solar drying works in dry regions, but is less reliable. Sorting removes visibly damaged or discolored kernels, reducing contamination in batches. Fungicides like propionic acid are applied to stored grain. Irradiation with gamma rays or electron beams kills mold and reduces mycotoxins, but consumer acceptance is low in some markets. The technology is effective, approved by FAO and WHO, but labeled as irradiated, which some consumers avoid. Ozone treatment is another method. Ozone oxidizes mycotoxins, degrading them chemically. Studies show reductions in aflatoxin and DON after ozone exposure, but ozone is reactive. Affects grain quality if overused. There's a final twist. In 2024, researchers at the University of Natural Resources and Life Sciences, Vienna, published findings on bread mold that challenged assumptions. The study used multispectral imaging, DNA barcoding, high-performance liquid chromatography with mass spectrometry, metabolite profiling. They intentionally inoculated 5 types of bread with common mold species: Paccalomyces variotii, Penicillium citrinum, Penicillium chrysogenum, Aspergillus niger, Chitomium globosum. They tracked mold growth, mycotoxin distribution, metabolite migration. The results confirmed that most mycotoxins stay localized. Fumonisins, Roca14C, viriditoxin, dihydrocitronone, all concentrated at the inoculation site and directly below. Lateral diffusion was minimal at 3 centimeters distance, but citrinin was the exception. It appeared throughout the bread, high concentrations in every tested alicate. The reason is molecular. Citrinin is smaller, more soluble, migrates through the porous bread matrix easily. Other mycotoxins are larger, less mobile, bind to bread components. This discovery has implications. It means that for most molds, removing the visible contamination might reduce risk, but for citrinin producers, it's unsafe, and you can't tell without testing. Home consumers don't have HPLC-MS. They rely on visual inspection. The guidance becomes conservative. When in doubt, discard. The potential exposure to citrinin, even in seemingly clean parts of the loaf, outweighs the cost of wasting food. The researchers suggested that partially moldy bread could be used for insect farming. Mealworms and other edible insects can tolerate some mycotoxins. Growth is impaired at high concentrations, but at low levels, insects survive. Feeding moldy bread to insects, then using insects as animal feed, could reduce food waste while maintaining safety for livestock and humans. The insects act as a buffer, metabolizing or sequestering some toxins. Research on this approach is ongoing. European regulations are evolving. Insect-based feed is gaining acceptance as a sustainable protein source. But for now, bread mold remains a household hazard and a scientific frontier. Every fuzzy patch is a colony of thousands of individuals, each a complex organism with genes, enzymes, metabolic pathways. They communicate chemically, compete with bacteria and other fungi, produce compounds that alter their environment. They've been doing this for hundreds of millions of years. Long before humans baked bread, fungi were decomposing organic matter. We built our civilization on agriculture. And molds followed. They infested granaries, contaminated stores, challenged food security. Ancient Egyptians stored grain in sealed silos. Romans used sulfur fumigation. Medieval Europeans lost harvests to ergot and fusarium. The Industrial Revolution brought understanding. Pasteur demonstrated microbial spoilage in the 1860s. Koch developed culturing techniques. Fleming discovered penicillin. The 20th century added molecular tools, genomics, metabolomics. The 21st century brings climate change, global trade, new challenges. The bread on your counter is a microcosm. The air contains thousands of spores per cubic meter. They land, germinate, grow. Species composition depends on geography, season, building ventilation, outdoor weather. Rhizopus dominates in warm, humid conditions. Penicillium in cooler climates. Aspergillus in dry, hot regions. Fusarium comes from fields, persists in flour. Cladosporium is ubiquitous. Neurospora is rare now, controlled by bakery hygiene. Monascus is incidental unless you're fermenting red yeast rice. Each genus has its niche, its growth requirements, its toxins. Together they form the moldy bread ecosystem. And the final cold fact, a 2024 analysis of bread samples from 5 types: wheat to rye oat, sourdough toast, white wheat toast, sliced wheat rye, whole loaf, wheat rye, gluten-free corn, showed mycotoxin presence in all preservative-free samples within 10 days at 20 degrees Celsius. Citrinin migration was confirmed in 90% of Penicillium citrinum inoculated loaves. The mycotoxin reached undetectable to trace levels only in toast breads, with calcium propionate and sodium acetate added at legally permitted concentrations. The study concluded that natural preservation methods, sourdough fermentation, salt content, acidity provide some protection but are insufficient alone. Chemical preservatives remain necessary for commercial shelf-stable bread. Consumer preference for clean label conflicts with food safety requirements. The industry navigates this by using minimal effective doses, transparency in labeling, and education about preservation purpose. The alternative is shorter shelf life, more waste, higher cost, or higher risk. You look at moldy bread differently now. It's not just ruined food, it's a battlefield of competing organisms. A chemical factory of toxins and antibiotics. A reminder that fungi are everywhere, waiting for the right conditions. They're patient. Spores survive for years. When moisture and nutrients align, they grow. And in growing, they reveal the complexity of life at microscopic scale. 7 species mentioned here, dozens more exist. Each one a story of evolution, adaptation, survival, each one producing compounds we've barely begun to understand. The bread mold you see is only the beginning.